Right, okay, so we've started to talk about biodiversity, and before we do anything else, we need to be aware of the definitions of the three levels of biodiversity. The biggest being differences in organisms due to their habitats, differences in organisms due to differences because they are defined as different species, and then finally, even within one species, we have genetic diversity, yeah? So organisms are different from each other because they have differences in their genes. Maybe due to mutations, maybe just due to sexual reproduction and different combinations of genes. Okay, so then the next idea is this idea of measuring biodiversity. So that's what we start off with. This is not the same pen that I was using. Measuring biodiversity. Okay, so this is ultimately just about sampling, right? So if we have biodiversity, differences in our organisms, how do we measure it? Okay, so very generally, this is about sampling, and what that encompasses is A, why we need to sample. Okay, so essentially that's about, if we want to measure the biodiversity of a forest, we can't go around counting every single organism in that forest, every single tree, every single bee, bird, etc. So what we do is we sample a small area and then multiply that up to account for the larger area. We're happy with that? Okay, so that's the why. Secondly is how. So what is it that makes a good sampling? And the two aspects of that is what makes that sampling random and what makes it representative. Okay, so I'm not going to go into details here, but we're just trying to look at what the broad things about the topic are. And measuring biodiversity is about sampling, why we do it, how we do it, how do we make our sample random so that we are not introducing bias. Yeah? So I'm not throwing my quadrat like this, I'm actually making a grid with numbers of a particular area, and I'm generating random numbers to tell me where I'm going to sample. Yeah, I'm not going over to that tree because it looks interesting. Yeah? And how do I make it representative? Yeah? So if I go out during winter to count the number of flying insects, the number that I'm going to sample is going to be low. But if I sample the same area in the summer, it's going to be high. So I need to make sure that I'm sampling at different times of the year. I'm using consistent methods. Yeah? Someone uses a sweep net down here, someone else uses a sweep net up there. It's going to lead to differences in what they collect. Yeah? So how do I make it representative? And the third aspect of sampling is which techniques you are using. Are you using a quadrat? Are you using a transect? Are you using a fly trap? Yeah? And that all comes down to the organism that you are sampling. Yeah? So if you're sampling plants, you're going to be using quadrats or the needle point frame. Yeah? But if you're sampling animals, you'll have to use different techniques. Okay? So essentially that's the whole of the sampling. That comes under that. And with the sampling, you're going to get some results. Okay? And those results will tell you two things. The richness of the area and the evenness of the area. Do you know the definitions? If not, you got to find out. Now, richness is about the number of species, and evenness is about the distribution of that species. How many species are there? Is one species dominating? If you take those two things together, you get the Simpson's Biodiversity Index. Is this making sense? Yeah? Okay? And once you have that, it gives you an idea of the biodiversity of this area using the techniques that you've used. Very good. Using the techniques that you've used to give you a quantifiable number that tells you what the biodiversity of the area is. Okay? 1 being high, 0 being very low. All right? That's what you should get at the end of that. Okay? So, after doing all of that, you get an idea of the biodiversity of an area, and that's sampling. That's basically that first quarter of the unit module done. Any questions? Too shy to ask.
If it's exactly 0.5, is the biodiversity high or low?
Good question. Good question. On its own, it's neither high nor low, but you might find that you're comparing the biodiversity of two areas.
Okay.
So in that case, how you interpret it will come down to what's the biodiversity of the other area in comparison.
Okay.
Yeah. So that's a good question. So 0.5 on its own is neither high nor low. However, in comparison to something else, it might be high or low. So if you're comparing it to another area that has a biodiversity of 8.8, then in comparison it's low. And if you're comparing it to somewhere else that's got 0.2, then comparatively it's higher. All right? Okay, so that is measuring biodiversity. Okay? I think my fans like a bit of real world ingredients in the videos. Right, so once we measure biodiversity, we have an idea of how, the different range of life forms or organisms. Then we classify. So what we're doing is once we sample, part of that sampling also involves identifying organisms. And maybe if in our sampling, we encounter something new, we might have to classify that organism according to groups and other organisms that it's related to, or maybe it's a new species. Okay? So, next, what we look at is classification. Classification. So the first one was measuring biodiversity, and next we kind of make sense of biodiversity by putting it into groups. And essentially that's what classification is. So again, we're going to start off with some key definitions. What is biological classification? Anyone?
Grouping of organisms into groups based on their observable features.
Yes, okay, so grouping organisms based on similarities or differences in their characteristics, that's biological classification. Okay, and on its own, it seems like a bit of an odd thing, it doesn't really fit in anywhere, but if you start to group organisms that you find based on similarities, you start to form a structure. Yeah? And if you look within those groups that you have, I'm going to put all the eukaryotes together, and then if you look at all the eukaryotes, you might say, I'm going to put all the plant ones on this side, all the ones with chlorophyll on this side, and I'm going to put all the animal ones on this side. And you further subclassify. All those animals with backbones I'm going to put here, all the ones without I'm going to put on this side. And if you carry on like that, you start to get a structure developing, and that structure is sort of the evidence for evolution, isn't it? The fact that you go through all these groups and they get more and more similar to each other. Is that making sense or not?
Yeah.
Okay, so it's linked to the next bit. So if you classify, it tells you, it gives you information about the relatedness of organisms. So, important definition, taxonomy is essentially how we classify. Okay? What do we classify based on? Am I going to classify these, am I going to group these organisms because these all smell nice and those don't smell nice? Or am I going to classify them based on these guys seem to have 2 leaves when they germinate and these plants seem to have 1 leaf when they germinate? How am I going to classify? On what basis am I going to classify? That is what taxonomy is. Okay?
Yeah.
Phylogeny is the other aspect of this, and this is what I was talking about with regards to evolution. Phylogeny is when you say, oh, these organisms seem to be more closely related to each other than those organisms. Therefore, I think that these guys must have evolved more recently and share a common ancestor, whereas they might not share a direct common ancestor with those organisms. If I'm using my groupings to get information about how related organisms are, that's phylogeny. In very simple terms, say this is a tree of classification, right? So I have those large group of organisms, and then within that large group of organisms, I've got these 2 groups of organisms that are very similar, and that group which seems to be different but still similar. Okay? Now, if I say, oh, shall I put my new organism that I have found from my sampling, shall I put it here, here, or here? That is taxonomy or phylogeny.
Say that again?
I found a new organism. Shall I put it in this group, that group, or this group? What's that? Is that taxonomy or phylogeny?
Taxonomy.
Correct. That is taxonomy. But if I say, oh, these organisms are much more similar to each other than they are to this group of organisms, that's phylogeny. Okay? So just bear that in mind. Okay. Now, as you classify, when you get your first main groups, you get, so I'm going to box that off. That's our first part of the discussion done on classification. Now, if you carry out the process of classification with all the organisms on the planet, you start to get 5 main groups. And they are called the kingdoms, okay? And what I'm not going to do is discuss what they are and go into detail of their features, because you can do that yourself. But you get the kingdoms, and as you go on further, if you look within the kingdom, you can further divide them based on characteristics. And as you look at those small groups, you can, even within that group, you can further subdivide. Yeah? And if you do that, you'll get the kingdoms, and then the phylum, classes, order, family, genus, and then finally species, yeah, which is just one group of organisms which are extremely similar to each other. Okay? Right, have I gone off the board? Family, genus, species. Kingdoms, what was it again? K, phylum, class, order, family, genus, and species. Okay? Right, okay. So that's the next bit. And this is how it was for a very long period of time until we developed new biochemical and molecular biology techniques. This allowed us to look more closely at the proteins of the organisms at their biological molecules such as DNA, RNA, and compare the sequences of bases, compare the sequences of amino acids to give an idea of how closely they're related, i.e., not just this organism looks similar to that one. Let's have a look at its biological molecules. Do they have the same sequence of amino acids, or are they slightly different? If we use that information, we further then subdivide, not further subdivide, but we reorganize our classification system into domains above the 5 kingdoms. Okay?
Oh, how many domains are there?
Yeah. All right. So 3 domains. So based on new biochemical and molecular biology evidence, we have 3 domains. We now group the archaea or archaebacteria differently from the bacteria, which are again different from the eukaryotes. Okay? And the eukaryotes, within that big domain, we still have all the other kingdoms, such as fungi, plant, protoctist, and animal. Okay? And that restructuring is because we had new evidence on which we could classify. Okay? And I believe that is classification. I thank you. So, if we look at this, what happens is we have all these organisms and they fit into 3 domains. Okay? But even within each domain, so if you have the eukaryotes, for example, they have 4 kingdoms within them, and within those 4 kingdoms, they have subgroupings, and they have subgroupings, and they have subgroupings, and they are further subdivided until finally you get a species. Unique. Okay? But what this structure tells us is that there must have been a process that gave rise to all these different species. If we go backwards along this tree, it's basically like going back in time. And if you go back in time, you will find that the number of different species essentially might get fewer and fewer, and you might find that if there's 3 species alive today now, they seem to have come from originally 1. And at that level, there might have been 3 different groups, which all might have originated from 1. So if we look backwards in time and then look at the process, how these species came to arise, you know, the undeniable conclusion is that they must have evolved, gradually becoming different from each other over time to become these different species. Okay? And that's the next topic. So I hope you can see now how classification is related to evolution. Looking at the classification, looking at the tree of life as it were, it kind of in itself is an evidence for evolution. And we only have that tree of life because we classify what we find. Okay, so the next thing is evolution. And this is a really important concept, okay? You'll find it cropping up in many aspects of F212. So it's really important that you understand this. Just like it's really important that you understand all your biological molecules. It's really important that you understand protein synthesis. This is another one of those key ideas, evolution. So evolution is highly related to biodiversity because evolution always needs, as one of its main starting ingredients, variation. Okay? And the particular kind of variation that we are talking about, sorry, it's not that one. It's this one. No, not that one. This one, finally got there. Genetic biodiversity, all right? There's differences within organisms that can be passed down to their offspring. Yeah? If you got green eyes because you got contact lenses, that's not going to be passed down to your offspring, is it? Because it's not a genetic trait. But any variations that are due to your genes, that is going to be passed down to your offspring. And so we need to just discuss very quickly, or just mention, that there are two types of variation: continuous and discontinuous. Is that ringing any bells? Yeah? So continuous and discontinuous. And remember that continuous variation shows a very specific set of characteristic features, such as not having intermediates, such as showing a pattern to the distribution, having very few at the extreme ends, but many in the middle average, and so on. And discontinuous, remember, doesn't show any sort of pattern. It has data that is separated into specific characteristics with few intermediates. Okay? So these are the features, but remember also what causes that variation. Continuous variation caused by the interaction between the genes and environment, or many genes. So many genes or genes plus environment. And discontinuous variation usually being controlled by either one or a few genes only. Is this making sense? Okay? Right, so that's variation, all right? Now let's move on. How is variation linked to change over time? Variation is change. Variation causes change over time because of this one thing called selection pressure. Okay? If you've got variation, that's not going to lead to change over time unless you add a selection pressure. So variation plus selection pressure is needed to get adaptation. And once you have adaptation, that means that your next generation is slightly different to your previous generation. And if the following generation is slightly different, and the following generation after that is slightly different, over many generations you're going to get change in the organism. Making sense? Okay. All right. So what do I mean by selection pressure? Selection pressure is changes in the environment. Yeah? Or any kind of challenge to an organism that causes a struggle for survival. So next to selection pressure, I'm just going to put in brackets, struggle for survival. Now, if you have a struggle for survival, what that means is, if you have lots of different organisms, and they're all different from each other, okay? A struggle for survival means that only the best adapted ones, only the ones with the right variations will survive. So say this one has got the right adaptation to survive, and all these circle ones survive. And all the ones that don't have that particular feature will not survive. Okay? Now this is why it's important there's a struggle. If there's no struggle for survival, they all survive. Yeah? And the next generation is pretty much made up of the same mixture of genes as the previous generation. But if there's a struggle for survival, that means that some of them are not going to make it. And the ones that do make it get to pass on their genes to the next generation. Which means the next generation are all, according to my diagram, they're all circular. And now we don't have the squares and the triangles and the rectangles. Yeah? And so we have change over time. Making sense? Okay, so variation continuous and discontinuous, is linked ultimately to this idea of genetic biodiversity. All right, so don't be surprised if biodiversity is linked to evolution in any question, because ultimately, if the more biodiversity that you have, the more variations you have within populations, within habitats, the more likely that that population can survive a selection pressure. Yeah? If all of these were the same and I impose a selection pressure, they all might not survive, or they might all survive. But if there's lots of variation, if they're all different, then there's more chance that one of them has got the right variations to survive. Okay? So, any variation that leads to a survival advantage is called an adaptation. And this is best exemplified in xerophytic plants, which overlaps with F211. Yeah? So the adaptations that allow plants to conserve water are all examples of little changes that occurred in species over time that gave them a better chance of survival. Yeah? So there might have been one generation of plants that didn't have any stomata. Yeah? But the few that did survived. The ones that didn't survive, dead, gone. So now all the ones that do survive all have stomata. Making sense? That's an adaptation. Okay, so if this process carries on for many generations, that gives you change in species over time. And ultimately, if you get to a point where your latest generation of organisms is so different from the original set that they can't breed and produce fertile offspring, you have a new species. Yeah? So it's just extending the idea of many generations. All right? So if I do many arrows like this, eventually you'll get evolution of species. Or actually, I should say, speciation. All right? So that's the essential ingredients of evolution. I know speciation comes into that, but maybe that's, yeah? Yeah.
Around how long would it take, like, selection pressure to completely get rid of, like, a species?
We are talking about very small changes from one generation to the next. So for a new species to develop, we are talking thousands, possibly even millions of years, depending on the life cycle of the species. So, for example, bacteria, they reproduce very quickly. So they can go through many generations in a short amount of time, and therefore speciation could happen quicker. But for humans or other organisms like that, we take a long time to reproduce the next generation, and so evolution can take much, much longer. Yeah? Right. Evidence for evolution is given by fossils, yeah? And you need to know what are the gaps in our information because of this, as well as what information they give us, right? But the basics of fossils is that as you look in the layers of rock in the Earth's crust, the deeper down you go, the further back in time you're looking, and therefore, according to our example, you might get change in species over time. So if you look in the same place and you're getting subtle differences in the fossils that you're finding, the deeper you go, it's showing that there is a change in the features the further back in time you are going, and that's the key point here. Yeah? That as you're looking back in time, there's changes in features, differences. Yeah? Not that they are similar, but they are different. And also, with more recent evidence, again, the sequences of proteins, the amino acids in proteins, the sequence of bases in DNA, that can also give you evidence for evolution. So, biochemical evidence is also incorporated into that. Okay, so you might be comparing the sequences of amino acids in an organism compared to another one to see how related or different they are. Yeah? Okay, and that, I think, is evolution. That brings us then to maintaining biodiversity, and the way I think of this as fitting into the whole context here is evolution is the reason why we have all this variety of life, one organism changing, maybe diverting into two species, those two species further adapting, evolving into more. And so evolution has given us all this biodiversity, and now it's the kind of human's responsibility to maintain that biodiversity for a number of reasons. Okay, for a number of okay? So these are some of the reasons why, and they could come up as like a suggest question or something. But how we go about maintaining that biodiversity, so there are ex situ conservation, which is taking organisms out of their natural environment in order to protect them from some danger that is causing them to be endangered in the first place. So they need to be removed from their environment, and that obviously has some advantages and disadvantages. And we also have in situ conservation as well. And that's keeping organisms in their own natural environment, but that has its own advantages and disadvantages also. Okay? So know the advantages and disadvantages in each case. Alright? And finally, the three organizations: CITES, Convention on Biodiversity, and the Environmental Impact Assessment. What helps in this case is to know that these organizations each have their own individual thing that they focus on. So don't just think of them as a group of organizations that are just maintaining biodiversity. They all have a specific thing they focus on, know what that is. And finally, that they are all international. So they all rely on some level for international cooperation. Okay? So the CITES is ultimately about trade in endangered species. Am I going off the board? Well, just about. Convention on Biodiversity is about maintaining, so each country's own responsibility to maintain, so making sure that each country is taking part in ex situ and in situ conservations, seed banks, exchanging embryos, botanical gardens, zoos, etc. And finally, environmental impact assessment. That is about making sure that any new development doesn't affect biodiversity too greatly. Yeah? Okay? So that's what the EIA is all about. And you might find that an EIA is often linked to a sampling, because you have to sample an area if you want to find out how biodiverse it is, if you want to make an environmental impact assessment. Okay? Right. So that's it. Hope you found that useful. An applause wouldn't be particularly